Hello, my name is Ahmed Khan, and I'm a PhD student at the Neuroinformatics for Personalized Medicine Lab at the Montreal Neurological Institute. Today, I'll be presenting some results from our project which show that neurotransmitter receptor mechanisms in personalized neuroimaging models can help explain the clinical heterogeneity of Parkinson's disease. I have no disclosures. Parkinson's disease is primarily a movement disorder characterized by its motor symptoms of tremor, rigidity, and the slowness of movement. Its main pathophysiological hallmarks are the loss of the dopaminergic system and the accumulation of alpha-synuclein, a protein implicated in neurotransmission. Despite being the second most common neurodegenerative disease, its causes remain unknown and there is currently no cure for Parkinson's. Parkinson's can be a complex condition to manage clinically. Its motor symptoms are typically treated symptomatically with dopaminergic therapy. However, this often comes with side effects or non-response. In addition, there are a variety of non-motor symptoms that are present for most Parkinson's patients in some combination. These often begin much before the motor symptoms and can continue to develop in severity as the disease progresses. It's clear that Parkinson's involves more than just the dopaminergic system, with other neurotransmitter systems often being targeted pharmacologically for specific non-motor symptoms. In addition, there is a selective vulnerability of certain neuronal populations based on neurotransmitter expression. This suggests that there might be multiple neurotransmitter-driven disease processes underlying Parkinson's. For example, the dual syndrome hypothesis of the cognitive impairment in Parkinson's suggests that executive impairment may be largely due to dopamine, while dementia and visuospatial dysfunction might be due to cholinergic degeneration in Parkinson's. However, a systematic understanding of neurotransmission dysfunction in Parkinson's has been limited due to the lack of receptor maps in a patient population. This is largely due to either the lack of in vivo radioligands for various receptor types or the expensive PET imaging for those receptors for which we do have radioligands. On the other hand, we do have longitudinal multi-modal neuroimaging datasets in Parkinson's, as well as post-mortem receptor distributions in the healthy brain. What this project proposes is a model-based approach to try to infer mechanistic alterations involving neurotransmission by combining healthy receptor templates with individualized neuroimaging data. Specifically, we do this by modeling the alterations to various neuroimaging modalities in combination with receptor densities and a structural connectome as templates. We then take these individual model weights as proxies of biological mechanisms and correlate them with clinical symptoms across patients. This receptor data consists of the average densities of 15 neurotransmitter receptors obtained from postmortem autoradiography. This covers 95 subcortical and cortical regions defined by cytoarchitecture, as well as 6 neurotransmitter families, including glutamate, GABA, acetylcholine, adrenaline, serotonin, and dopamine. We combine this with neuroimaging data from PPMI, consisting of structural, functional, diffusion, and dopaminergic SPECT imaging, as well as a tractography template from the Human Connectome project. We modeled the longitudinal changes to each imaging modality as a combination of local effects and the network propagation and pathology in a dynamical systems framework. Specifically, these local effects are the receptor-mediated interactions between different imaging modalities at each brain region. We find that regional neurotransmitter receptor expression helps explain the longitudinal changes to each neuroimaging modality, although this is more pronounced for certain imaging modalities, namely gray matter density, T1-T2 ratio, and mean diffusivity. Across all imaging modalities, we find important and significant interactions between imaging modalities at each region that drive their longitudinal evolution. We also find that the receptors driving susceptibility to degeneration are dependent on the type of imaging modality. For example, we find that cholinergic mechanisms are important for predicting gray matter atrophy, while fractional anisotropy is more dependent on kainate and AMPA receptors. In order to identify the clinical correlates of these model-derived mechanisms, we looked at various clinical assessments from PPMI, which cover motor, non-motor, psychiatric, and cognitive domains. We then linked our model-derived biological mechanisms with these clinical symptoms using a partial least squares SVD decomposition into latent spaces. After testing for robustness, we find two significant latent components representing about 48% of the variance and about 13% of the variance respectively. There is a strong correlation between the latent projections of clinical scores and model-derived biological mechanisms in both these components. And specifically, they seem to correspond to the motor symptoms and psychomotor speed in the case of the primary component, while the secondary component largely corresponds to visual spatial function, psychiatric symptoms, and memory. In the primary component, we note that there are strong GABAergic and glutamatergic mechanisms, specifically acting via pathway affecting functional activity, mean diffusivity, and T1-T2 ratio. In comparison, the second component has more cholinergic effects as well as glutamatergic effects. This kind of model-derived inference of underlying molecular mechanisms can be used to guide personalized and precision medicine. For example, identifying different therapies that might benefit different kinds of subjects. Applying a similar analysis to Alzheimer's disease, where we compared model-derived receptor alterations with a normative healthy control population, we found that subjects with similar clinical profiles might have different underlying receptor alterations based on their neuroimaging changes. However, the lack of control subjects with sufficient data in PPMI prevented a similar kind of analysis in our current cohort. This requirement of longitudinal and multimodal data would be a limitation for clinical applications. We would also need to validate our method and our results for robustness to different parcellations. Of course, the interpretation of these imaging modalities as specific forms of pathology is also not that direct. And we would also need to validate this method in terms of response to different kinds of treatment clinically. To summarize, our model-derived receptor mechanisms help explain neuropathological alterations in Parkinson's disease. We find two overlapping disease axes in which model-derived mechanisms correspond strongly with clinical symptoms. Specifically, motor symptoms and psychomotor speed associate with GABA and glutamate receptors, whereas visual spatial function as well as psychiatric symptoms and short-term memory are associated with cholinergic and glutamatergic mechanisms. Our methods will be available in our lab's open access toolbox very soon. And I'd like to thank everyone involved in this project, especially my PI, Yasser Oturia Medina, as well as our external collaborators, including Nicola Palomero Gallagher, who collected the receptor data. Thank you.